So we went through a few lectures that touched on gene expression and microfluidics, and today we continue along this line. So today's speaker is Attila Becskei. Attila has been working on this topic ever since his PhD, when he was working in EMBL, and he published one of the very first artificial networks in a negative feedback loop with Serrano. This is really a fundamental paper. Then he went on to work on a similar topic in the US in the lab of Van Ordenauer, and then he came back to Europe in Switzerland, first in Zurich and then in Basel, where he is now a full professor. And so I give the stage to Attila.
Okay, thank you very much. I'm happy to give a talk. I think this would be the first. I had several years ago a talk in Hungarian, which did not really work out because I spoke in Hungarian and some people criticized some of the translations. So at least you cannot. There will be no means for criticism because I will talk in English. So I will talk about stochastic gene expression and feedback control. These are two large areas in systems biology. So this will be just few glimpses, and you will also hear about why these two things are related. So first, I will talk about stochasticity. Stochasticity is it can be used synonymously as for randomness, but it's also essentially represents one way to model quantitative biological processes. So there is, I think, a relatively widespread misconception about stochasticity, because some people may think that these are random events. I just want to say that as it's very unlikely that there are truly random events. So the first main discussion in the area of mathematics and philosophy was started by Laplace, who claimed that if there is a demon who knows everything about the universe, and then at that time physics was already reasonably developed, and then he claimed that we can predict all the future position and events of everything in that universe. So one very important thing is, if we believe that world is deterministic, then we can predict everything. So it's all about the exact predictability. And so I really don't want to go into details of whether world is deterministic or not, but at least much of the physics claims that it's deterministic. I would like also to say that determinism also exists outside of science, like there is religious determinism, which is called predestination. And some people claim that there are quantum phenomena which are not really deterministic, but at least in our macroscopic world at the level of cells and so on, we don't really think that quantum phenomena are playing a major role. So what I try to say, that determinism is not only a worldview, but it has an important implication for modeling, because it means that if we know everything, of course, this is already a very strong statement that we know everything, but of course this is it's a hypothetical statement. Then we would be able to to model exactly how things work. Of course, many things appear random. For example, if you just have a single a billiard ball, then you hit it, then you can exactly predict its movement. But if you have 6 balls and then they collide at the same time, then it will be more and more difficult to predict. So it just shows that randomness appears simply due to the increasing complexity. So what you will be able to see is kind of deterministic and stochastic models have been there in all areas of biology, maybe not using this name. So for example, if evolution had 2 main theories, one is Lamarck's theory, and Lamarck claimed that if there is a stimulus for an animal, for example, if an animal has a short neck, and then if it tries to stretch its neck to reach the leaves, the constant stretching of the neck will change the genetic material, which will be then passed off to the offsprings. On the other hand, Darwin claimed that there is a distribution of traits, and only certain animals which are a bit taller, they will be selected, because, and that's how the distribution of the genetic distribution will evolve over time. So and so you can see that in some way the Lamarckian evolution is in some sense a deterministic evolution, while the Darwinian evolution is in some sense a stochastic evolution. And then you will be able to see that that it's even the random events can be modeled, just we are not modeling precisely each event, but we are modeling the distributions. We are modeling the probability distribution. So in microbiology, the people may have assumed that Lamarckian evolution is still a valid assumption, and then this was proven by Luria and Delbrück by a so-called fluctuation test, which very nicely illustrates what stochastic modeling is. So for which they got a Nobel Prize a couple of years later. So, well, in fact, not a couple of, but 20 years later. So what they did is they took plates which had phages, in fact, because bacteria can evolve resistance against phages, but this is something similar, that as if there were only antibiotics in the plates, and then what is important, whether bacteria develop resistance against antibiotics. So if you assume that so the experiment was done that there were several flasks, microbiological flasks or tubes, and in each tube they cultured bacteria for several days, and then they plated these bacteria. So you have plate 1, plate 2, plate 4, and each plate is assigned to a culture which was cultured for several days previously. And if you can assume that if the resistance is induced by the phages or antibiotics in the plate, then there is more or less equal probability of bacteria, resistant bacteria arising. So which means that on each plate, some bacteria should appear with relatively similar number. This is kind of an extreme case where you see that it's more or less 2 bacteria per plate, but this is a bit ideal. So there will be still some distribution. On the other hand, if the mutations are spontaneous and not due to the adaptation on the plate, then mutations can arise very early during growth or very late or not at all. And then consequently, you will see very different numbers of bacterial colonies on the plate. On some plates you will have nothing, in some you may have thousands or one or two, so which means that the distribution will be extremely broad. This is the so-called Luria-Delbrück distribution. This is in fact even a research area in mathematics, because it's a very non-trivial distribution. There is no even explicit form for this. While this one is more or less a relatively well-defined distribution, which is most likely to be similar to the Poisson distribution, even though you see that only here is not even a distribution because it's really two bacteria, but in reality is more similar to a Poisson distribution, which is much narrower distribution to the Luria-Delbrück distribution. So what is important that this was observed in the experiments, which showed that it's the Darwin's evolution works also on bacteria. And what is also important that it shows how stochastic models can be used. So the stochastic model is not predicting exactly that, whether on this plate 2 or 3 bacteria will survive, but it simply predicts a distribution, and then you can compare two different distributions. And then, because they observed this very broad distribution, this showed that it is a Darwinian evolution. So this is the summary of what I said before, that in contrast to deterministic predictions, where you can make very precise statement about every outcome, you can make also stochastic models, which it does not tell you precisely the outcome, but it still gives you distributions. So this was the evolution, but this is also widely used in, let's say, in statistical physics. For example, the Maxwell-Boltzmann distribution and so on. So, and what I'm trying to say is that stochastic modeling does not mean that things are random, because also the mutations ultimately appear due to some replication errors, which again is some very precise process, but we don't care all the details. We just want to see how the differences in the assumptions lead to different distributions. So now I will switch to stochastic processes in gene expression, and you will see there that probability distributions are also very useful there. And you can also make a lot of simplifying assumptions about probability distributions. So one of the most common way to characterize the distribution, so the first two moments are the average, and the second is the standard deviation. And then it's quite useful to normalize them by dividing the standard deviation by the mean. And then you get the so-called coefficient of variation, which is also a very useful way to measure the fluctuations in gene expression, which we also call noise. So a very important the coefficient of variation is really much beyond noise. It can be even used to compare the measurement errors of different techniques. So for example, if you measure the distribution of the measurement error of glucose in the blood or erythrocytes, what is important that these are non-dimensional numbers or can be expressed as percentages. So the coefficient of variation of 2% means that on average the error around the mean is 2%, has a 2% probability. Or 2% of the mean value is the average measurement error. This is what is important. You can very easily see that it's non-dimensional. For example, if you do many measurements are done by fluorescence, and then the fluorescence intensity simply influences the absolute number of the measurements. So for example, if you have 1, 2, 3 molecules of GFP in a cell, then if you have a weak laser, then you get 1, 2, 3 arbitrary output. But if you have a stronger illumination, then you get 10, 20, 30. If you calculate the mean and the standard deviation, you will see that you will get exactly the same coefficient of variation independently of the illumination. On the other hand, if you use other measures, for example, the Fano factor is something similar, but there you don't divide the standard deviation by the variance by the mean, because many people divide the variance by the mean. And then you can see that this normalized factor, the relative variance or the Fano factor, will be different. It is 1/3 here and 10/3 here. So this shows that not every measure is dimensionless. So this coefficient of variation is very useful, and you will see that I will give you a couple of examples to quantify the distributions with this. So now the second question arises why gene expression is that process in molecular biology or biochemistry or cell biology which is particularly stochastic, or why there are broad distributions. And the answer is that this is due to the law of rare events, which is simply due to the fact that genes are present at low copy numbers. So each diploid organism contains 2 genes, and depending on the cell division, there could go up to 4 or so on. The second thing is also RNAs. RNA molecules are typically present at low copy numbers, maybe 1, 2, up to tens, or sometimes it's even 100, but it's relatively low copy number. And you will see that why the low copy number is generating noise. And then secondly, you will see that how feedback loops interact with gene expression. So some types of feedback loops, especially the positive feedback loops, can amplify noise, for example. And then there are some potentially other sources of noise, like chromosomal loops and so on. I will also talk about later. So the small number effect was proposed by Poisson, again, nearly 200 years ago, and that can be simply explained by the following thing. Imagine you have a single molecule in a cell. If you want to increase the concentration, the only way to go from 1 is 2, but that's a huge change. It's 100% changing in relative terms. On the other hand, if you start from 10, the next number is 11, that is only 10% larger. So which means that you see that there's 100% difference from 1 to 2 versus from 10 to 11, it's only 10%. This also clarifies why the noise, as defined by the coefficient of variation, is inversely proportional by the copy number of molecules. So if you have one molecule, then this intrinsic noise is 100%, whereas if you have let's say 100 molecules, then 1 over 10 is 10 times lower noise. So, and then as I said earlier, the genes are always present at low copy numbers, but also often mRNA molecules. So that's why, because gene expression links DNA to RNA, gene expression is a particularly noisy process, as opposed to biochemical processes, let's say in glycolysis, when an enzyme converts glucose, where there are millions of glucose molecules or in a cell, or at least more than 1,000 molecules. This is typically not so stochastic. So as a rule of thumb, you can say that, so when is stochastic modeling really required? Well, this is really just a very rough number. I would say that whenever you have less than 20-30 molecules, then you should really do stochastic modeling. If you have molecules above 20-30, you can still do stochastic modeling, and stochastic modeling can be still relevant, but it's not a big fault if you already do deterministic modeling. You will see that deterministic modeling is always much easier than stochastic modeling. So it is you can gain much more easily insight with deterministic modeling. So there is an advantage of doing deterministic modeling. So now this is people have measured, in fact, gene expression noise simply by looking how, if you so people here tagged a gene with a green fluorescent molecule, and then they measure the distribution of the GFP expression in each cell, and the yeast genome has around 5,000 genes, so this roughly contains 5,000 different genes, and indeed you can see that there is an inverse relation between the noise and the abundance, which is very similar to this Poisson law. There is some change from this linear inverse power law relation because of some background noise and so on. But you can see that at least over a broad range of abundances, the linear relationship is quite clear between the log abundance and the log CD. So now I mentioned that
in that plot, each dot was a cell, correct?
Exactly. Oh, no, each dot is a gene.
Mm-hmm.
Each dot is a gene. So typically the dots here are ribosomal genes, which are highly expressed. And then for each gene, you do a microscopy or flow cytometric measurement, and then you simply measure the standard deviation of the distribution of the GFP molecules.
One cell can be measured multiple times.
So each dot is a measurement of, let's say, 1,000 cells.
Mm-hmm.
1,000 cells containing the same gene tagged with a particular GFP.
Mm-hmm. Okay.
Yeah. So these are typically here ribosomal genes, which genes expressing ribosomal proteins, which are highly abundant, while those genes which are here are typically transcription factors or other regulators, which typically have lower expression. And so, for example, here you can see that this abundance is real, so it's true abundance. So, for example, you can see that proteins, the transcription factors typically have copy numbers between 300-400 up to 1,000 copies per cell. Yeah, while ribosomal proteins have typically 10,000, or enzymes which are involved in glycolysis, they also have often copy numbers around 10,000 or even more. And they have simply lower noise than those. Yeah. So this is a relatively clear. So what I would say that the basics of stochastic gene expression were more or less finalized by the around 2005-6. So the basic laws of stochasticity were more or less shown quite broadly. So here I just would like to say why deterministic modeling is simpler. And even though you can have very similar processes arising, so for example, here there was Belousov and Jabotinsky in the 70s. They played with, or maybe even the 60s, they tried to reconstruct the citric acid cycle in vitro. So they combined some bromates with some alkali element catalyst and some organic acids. And then after a while, they started to see that some very interesting patterns arise from this. Nobody believed, because they assumed that only open systems can do this, while this is a closed system. And they did not believe that this is a physical possibility. And what you can see that these are similar to embryonic patterns. But the difference is that in embryonic patterns, there is a little bit more noise. So that would require maybe some stochastic modeling, but you will see so that deterministic modeling is much simpler. Deterministic modeling is done by differential equation, which is typically ordinary differential equation if the thing changes only in time, or partial differential equation if it changes both with respect to space and time. While if you want to model probabilities, then you have to, you can only assign a probability with respect to change. So a deterministic model will tell you exactly what happens in this point of space in over a certain period of time. While a stochastic model, which just gives you probabilities of changes, what is the probability that the concentration will increase or decrease to a certain degree. And first, for example, one of the simplest differential equation, ordinary differential equation, which can be also used to model gene expression, gives you that each RNA is produced by a certain rate, a production rate constant, tells you how much mRNA will be produced per time. And then you will have a decay with a decay rate constant. And then this gives a steady state solution, which is nothing else than the mean value of, that it gives you a certain level of gene expression. On the other hand, if you want to model the same with a stochastic model, then you have to write the probability for each molecule number. If you have 0 molecules, 1 molecule, 2 molecule, so each state has its own differential equation, then you have to convolute this. And then at the end, if you solve this, the corresponding master equation, then you get the so-called Poisson distribution. So this is one of the standard distributions in stochastic gene expression, because it will tell you that you will not have an exact number of molecules, but it will be a distribution, and the distribution is given by the Poisson distribution, where this lambda is exactly corresponds to the mean. So, which means that the mean value of a distribution is equal to the prediction of the deterministic model. Then we claim that there is no deviant effect, And in most cases, at least in linear models, the deterministic solution is identical to the mean value of the stochastic solution. I will show you later cases when this is not the case and deviant effects arise. So a classical case when deviant effects arise is the positive feedback loops. And when deviant effects arise, then it means that it's even not possible to model something deterministically. And despite the fact, yeah. So positive feedback loop is something very simple, but there are a lot of non-trivial outcomes. So it's essentially impossible to understand the behavior of positive feedback loop without mathematical models. So for example, if you model the elementary positive feedback loop, if a protein is encoded by a gene, and this protein happens to be a transcription factor which activates its own expression, then you have a constant production rate, then you have a production rate which depends on the concentration of the transcription factor, which here we describe by S as a substrate, and then this is also degraded. So most of the time when I ask the students what happens with what is the outcome, what is the positive feedback going to do, the typical answer is that you will have more expression. And this is true in a very simple case of positive feedback loop, because this is an autocatalytic term here, and this will increase the production. And there is this simple graphical approach that if you change S, you can plot the production rates and the negative terms, which is the decay rate, and their intersection will give you the steady state level of this protein. And then you can see that this will be higher with this autocatalytic term as if there was no autocatalytic term. And then the solution would be, let's say, 0.5, while now it's around 100. So there is indeed more protein expression. But what happens if the binding of the transcription factor to the promoter is not simply linear, but there are multiple binding sites in the promoter? Let's say it can be even just 2 or 3. Then the 2 transcription factors can interact on the promoter. So the binding of the first transcription factor can enhance the binding of the second. And if you have already learned about cooperative binding, let's say in cooperative binding of oxygen to hemoglobin, then you know that that results in a sigmoidal shape. And the sigmoidal shape will, in the context of a positive feedback loop, will create a very interesting phenomenon that you have not only one intersection, as it was here, but you will have 3 intersections. And out of these 3 intersections, only one will be an unstable state. I will have no time now to explain why is this unstable, but you just accept that this is the case, while 2 are 2 stable states. And these 2 stable states will essentially correspond to 2 different steady state levels. And this was to some extent something which I did during my PhD, that if you have a transcription, so this is a control system, if you have a transcription activator activating the expression of a GFP, you will see that all yeast cells have some expression. There will be some green color in all the cells. while you will, but if there is another change that the transcription activator does not only activate the expression of the GFP, but also its own expression, then you will see that the green color is stronger. Indeed, you see that it's stronger. So here's the autocatalysis. But at the same time, you will see that some cells are not expressing the green color at all. This is the so-called bistability, that there are two stable states. And it's impossible to predict which cell will become green or which will stay in the low state, but it creates two states, and this means that cells differentiate. People also suggest that that similar positive feedback loops are important in the cell fate determination in other developmental processes, like the expression of certain different types of photoreceptors in the retina, which is also controlled by positive feedback loop. So what you can see here, that the positive feedback loop amplifies stochasticity because you don't know in advance which cell will switch to the high state and which not. You can just model the distribution. And there is one interesting feature: the more nonlinear the system is, so what nonlinearity simply measures the degree of cooperativity. So for example, hemoglobin, when it binds to oxygen, and the nonlinearity is often measured by the Hill coefficient, the hemoglobin has 4 subunits. In an ideal case, the Hill number would be 4, but in reality, that's good. But in reality, in reality, there are 4. So the Hill coefficient is only 2.8, and this gives you the measure of nonlinearity. But typically, the stronger the nonlinearity, the more pronounced this bistability is, which means that if you change other parameters, you still get 2 states. While if you change some other parameters, here you just get 1 state. For example, here, well, here you get always at least 2 states. So the more nonlinear the system is, the stronger these unusual features become, such as bistability. And as I said, stochastic modeling is truly required whenever you have these strong nonlinearities, because it's very difficult to understand it with classical deterministic models. And here we, in fact, very often, so what was a bit unusual in this positive feedback loop, and this we did not fully understand, and very often people were surprised why these cells are not expressing GFP. So in fact, when we submitted this paper, we had very hostile, I still remember after 20 years, we had very hostile remarks on this, and then they claimed that these cells which are not expressing GFP, they are dead cells, even though we had even shown experiments where we showed that these cells, even the dark cells, grow and divide, so they simply cannot, so they are not dead cells. But I also had some feeling that it's an unusually strong, how to say, two states, a separation between 2 states, especially because we had just 2 binding sites for transcription activator where the cooperativity is not really that strong. And only later, so much later, we worked in here, which explained where this nonlinearity comes from. So in fact, we noticed that the synthetic networks, when you design a synthetic transcription network, is done with the goal that it's fully separated from the rest of the cells, and then it's very easy to model these, because in cells every interaction is hit, or many interactions are not characterized, so it's much more difficult to model. But this is also just an ideal assumption. Most synthetic networks do in fact interact with the cells, and we notice that if the expression of the protein is high, then the cell growth slows down a little bit. So in fact, the green cells are those which slow down. And what we did is that we introduced so-called stem loops into the RNAs. And these stem loops are those tools in the RNA which reduce protein translation. So you still have the same feedback loop, but with much less amount of protein. Then we still get the classical features of this positive feedback loop. We get two states, but we wanted to get rid of all nonlinearities. So in order to do this, we had a single binding site in the promoter, and then you can have nonlinearity also due to dimerization. And then if we fuse the two protein domains, there is no more dimerization. And then indeed, you don't have two states. And then here we change the binding by this molecule called doxycycline. The more doxycycline you have, the more binding.
What is on the y-axis on the plots on the right?
Sorry, it's just the fluorescence. Fluorescence, how strongly so this protein in addition drives the expression of a GFP. So this is then just a GFP. And then you have two different initial conditions. Either you start from a high initial condition or low, because we have another control which determines the initial state. So if you start from the, independently whether you start from the high state or the low state, you can get so the system will evolve to the same state. Yeah. So on the other hand, if we let proteins to dimerize and bind to multiple sites, you will get two very different states depending on whether you start from a high initial condition or low, and the cells will stay in the high state even after hundreds of cell division, which means that the system has a perfect memory, it's a perfect bistability. And then we also found that if you either have two binding sites and monomers, or you have a single binding site and the dimer, then you have still bistability, but it's a much narrower range. So here we have shown that it's not only the cooperative binding to the promoter, but itself the dimerization is sufficient as a nonlinearity to provide. But if you combine the two, dimerization and cooperative binding, and it's an extremely strong, it's a very strong bistability. And this is why the dimerization is particularly important, because dimerization is very widespread. More than half of the proteins dimerize, and so it's a very widespread. Most of the time, it's a neglected source of of nonlinearity. And here we have shown that even dimerization contributes to nonlinearity, and then subsequently to the bistability in the positive feedback loop. So, and then what I also wanted to say that, of course, that when you have only either dimerization of cooperativity, over time, the two states collapse. So this is simply because of the stochastic transitions between the two states. So one easy way to explain these multiple states is that it's kind of a potential well, and the cell state jumps out from one state to another one. But what is important that in positive feedback loop, it's essentially impossible to understand the realistic behavior of the cell without some stochastic modeling. And then what is also a very interesting thing that people earlier thought, partially due to our initial experiment, that you have always two states at the same time, but this coexistence of two states is partially due to the side effects of the network on the cell. But if you reduce the expression of the proteins, you get also more nonlinearity, and in fact then you get extremely strong bistability where there is no very minimal switch from one state to the other. So which means that with as simple as one single feedback loop with dimerization and cooperative binding, you have complete separation of the two states. And you don't need anything more complex. Of course, you can combine multiple feedback loops and you can make it, but what I'm trying to say that even with a single positive feedback loop, for all practical purposes, you can create very strong divergence of two cell fates. And of course, if you need multiple cell fates, you can combine multiple positive feedback loops. And then here I just want to say very briefly that whereas the positive feedback loop broadens the states because you create two phenotypes, the negative feedback has an opposite effect because it essentially, if you have a distribution of a gene expression, if you create a negative feedback loop, this distribution will be narrow, at least as long as the feedback loop is simple and fast and efficient. If the feedback is slow, it can even start to oscillate. Then you don't see this effect anymore. But at least in bacteria, most of the feedback loops are relatively simple. So this is another experiment which shows the same from the group of Balaji, and he has shown that even a bimodal distribution can be converted to a very narrow distribution by a negative feedback loop. So it kind of does the opposite to the positive feedback loop. And then, so this was kind of the basics. I don't know if it was basic or not, but this here, so far I gave an overview of stochastic gene expression and feedback loops. And here I want to discuss something which is we cover in our more recent research area, is the stochastic gene choice. So stochastic gene choice is another area where stochasticity plays a major role, and even biologists like it, which means that because you really need stochasticity to explain some of the phenomena, and it's here the stochasticity does not simply affect only the concentration, but even what gene is expressed. And the prototypic example of stochastic gene choice is the olfactory receptor. So what is very interesting that you may know that we have around 30,000 genes, and the largest gene family among these is the odorant receptor. So mice, but also humans, have 1,000 to 2,000 different genes only to encode odorant receptors. So each odorant receptor recognizes a certain odor. So unlike the taste receptors, where we have only 4 main tastes, the odors, there are a very large number. And what is interesting, that each neuron in the nasal olfactory epithelium is expressing only one specific type of receptor. So, which means that you will have always one neuron which expresses this odorant receptor, this, and so on. And which means that the cell has to ensure that no more than one receptor is expressed per neuron. And that is by far not so trivial, how to achieve that one odorant receptor is expressed, but not the others. This is more or less chosen randomly in the cell, and the others have to be suppressed. And it's not really understood how this is achieved. And what I wanted to say, that there is another family which is very similar to T cell receptor. There are many different T cell receptors which have a similar status, because the T cell receptor has to recognize different antigens, and also different T cells will recognize different antigens. And to make the complicated story even more complex, people have found another gene family which is a little bit similar to the olfactory receptor, is the protocaterin gene family, which are cell adhesion proteins, and they mediate the interaction of one cell to another. And there are different clusters of these protocaterins, so we are particularly interested in this alpha. And from this alpha, there are only, let's say, 2-3 isoforms are expressed. So not only one, but let's assume that from this region only 2. And this shows that it's still somehow exclusive, because one isoform is expressed from the maternal chromosome, and one isoform is expressed from the maternal. And so paternal and maternal gives you altogether at least 2 isoforms. It's still an exclusive expression. This is something you could imagine like the queen and the king jointly govern the country. So we have 2 isoforms, but it's still an exclusive society. And how this is achieved? So here we did single cell gene expression analysis, and then we differentiated embryonic stem cells through neuronal progenitors, and here you see neurons. And you can see that embryonic stem cells of protocaterins are not expressed, whereas in neurons, each cell, so each line is a cell, and then different isoforms are expressed. And if you count per cell how many isoforms are expressed, then we get on average 5. So again, we have a probability distribution. And then if we measure this distribution, so just as a brief summary, so you have cell 1, you can have 4 genes expressed in a cell, or 0, or 1 or 2. And then again, you can measure the variance. If the variance is very broad, then typically this is what we call co-occurrence. whereas if you have each cell is expressing only one or two, then the variance is much narrower. So again, we measure the width of the distribution, then we call this exclusivity. And we essentially checked how this variance changes over cell differentiation. So these are the early days of cell differentiation, and these are the later days. And what we have seen that the trajectory is something that the cells proceed from large variance and then they reduce their variance. So over differentiation, the distribution becomes narrower in the choice of protocatenaries. And what is important, so here the most critical thing was to find the correct distribution, because this is, here we don't have only a single gene, but we have number of genes, and each gene can have an on state and an off state. And this is what is called the so-called Poisson binomial distribution. So it's neither Poisson nor binomial, it's a Poisson binomial distribution. And then with this, you can measure the variance, and then you can compare the predicted variance based on no interaction between the genes versus the real variance. And based on this, you can decide whether you have exclusivity or co-occurrence. And we have seen that cells proceed to co-occurrence. So how this stochastic gene choice is working. So people have shown before that there are ring proteins called CTCF, which ensure that they bind to the promoter of one of the isoforms, and there is a downstream enhancer, and then they link the enhancer with the promoter through this ring-like structure, and in this way the particular isoform gets expressed while the others not. So that comes to the exclusion of the other isoforms. So this is the molecular mechanism of gene choice. And indeed, if we measure the CTCF binding by chromatin immunoprecipitation, we get a very strong linear relation with the expression frequency of the given isoform. And then we continued this a little bit further, and we wanted to ask, so there are these odorant receptors, there is protocatenin, and we ask whether this Poisson binomial distribution can be used to detect other gene families to provide cell identity. So why is cell identity important, and how many cell identities you need? So you can think of cells or countries. So you have, let's say on a map, countries. This just happens to be Central Europe. It's, I think all the relevant people in this audience are included: Switzerland, Hungary, Italy. So, and you may have heard of a mathematical theorem called X-color theorem. So if you have countries of any complexities, what would be your guess? How many different colors you need to distinguish all countries from the neighboring countries? For example, if you have just squares, already two colors are enough to distinguish a square from all the neighboring squares. So what would be your guess if you have complex shapes? How many colors would you need? Do you have any guesses? No guess. So you need, so earlier people thought that it's 5-color. So the 5-color theorem was already proven 100 years ago. With 5-color, you can color any map so that you distinguish any country from the neighboring countries. But later they have shown that even 4-color is enough. And you can see this is another case when it shows that mathematics evolves as well. because this was shown more recently. So you can see that with 4 colors, you can really distinguish any country from the neighboring countries, which means that you provide a cell identity, that you provide an identity to the country. And this is the same for tissues. If you have an epithelial tissue, and then you want to distinguish from a cell from the neighboring cells, then you can have 4. So why is this important? Because then you just need to have a family of 4 or 5 genes to distinguish yourself from the neighboring cells. Of course,
the genetic properties independent from the map.
Yeah. And of course, the situation becomes more complex if it's 3-dimensional. 3-dimensional requires more identities, but but this is still, many tissues are 2-dimensional, and with 2-dimensional 2-5 genes, and in fact many gene families are because not all, obviously the olfactory receptor takes already 1,000 genes. The protocatarins just take 12 genes, so they are not independent. There is no infinite number of genes. So we simply looked at single cell measurement of expression in a lot of different cell types. You may have heard that single cell expression can be measured. Single cell expression can be measured with this RNA-seq, and we were surprised how good quality there are some very good quality measurements, and we measured in very different cell types, and then we measured gene families in different cell types. And so first of all, we confirmed in a T cell receptor has a very strong exclusivity. So we quantify this Poisson binomial, and then if it's 1, then it's independent expression. If it's less than 1, it's exclusive. If it's more than 1, then it's concurrent. Most gene families are concurrent. We are not showing that. 95% of gene families are concurrent, but in most cells we found exclusive families. The strongest exclusive family is still the T cell receptor, which I mentioned at the beginning. The odorant receptor we don't show here. And then we also, was very interesting, that we found other families like synaptic adhesion expressed in corticostriatal neurons or other cells. And then we found even ion channels. So, but there is a certain number, or the ECAM, intercellular adhesion molecule, expressed in the prostate stromal cells, for example. And what we found, that there is a very strong enrichment of these gene families for cell adhesion. This is the most enriched cell function. And then this indeed explains that you need genetic diversity to provide cell identity to the cells. And how is cell identity defined? Primarily, this is defined to the neighbors. And the neighbors are most of the time defined through cell adhesion, or the other is interaction with foreign molecules, like in the odorant receptor or the T cell receptor. And what we were surprised to see, the second most enriched function is ion transport, which we still do not fully understand why. But it's also clear that in most cases, the two functions are not separated. You may have heard of the sodium-potassium ATPase, is very widely discussed in all introductory ion channel biology. But most people, and we also did not know, that there is this gamma subunit of this sodium-potassium pump, and each of these gamma subunits is having a sugar side chain, and they are in fact mediating cell-cell adhesion. The same thing is for lactate. So you may have heard of the lactate transporter, but is also involved in one of the subunit of the lactate receptor is also involved in cell-cell adhesion. And the same is true for carbonic anhydrase. Carbonic anhydrase is involved in ion channel transport, but it's also involved in the cell-cell adhesion in the brain. So this was this second work. I would like to acknowledge the work of Takeo and Sandrine in my group, who did most of this cell adhesion work. So, and here where I would like to finish. So in the introduction, I talked about why stochastic modeling is, or do you still see my presentation, or did I exit?
But it's okay, we can see it.
Okay, so I talked about stochastic modeling, that it already started with distinguishing what evolution takes place in microorganisms. That was the first, one of the first successes of stochastic modeling in biology. And then I mentioned that the stochasticity distributions are very important in gene expression due to the low number of molecules, what we measure by the coefficient of variation, and the Poisson law relates that the copy number of molecules is inversely proportional to the coefficient of variation. And then the feedback has a strong interaction between stochasticity because it amplifies noise, but the negative feedback can reduce stochasticity. And then in this second part, I talked about one specific biological phenomenon called the stochastic gene choice, when not only the state is chosen, but from a family of gene isoform, only one or a certain specified number of molecules is chosen, and where we found that there are other gene families in addition to the three classical families like the odorant receptor, T-cell receptor, and the protocaterins. There are other gene families, particular cell adhesion families and ion channels, which are also showing probability distribution, which is similar to the distribution of T-cell receptor or factor receptors. And I'm happy to answer any other questions.
Thanks a lot, Attica. It was very, very nice. So while people think about their questions, I ask you, so I really didn't understand. So what is the mechanism that makes sure for the whole life that gene is selected, for example, for this olfactory system?
So can you repeat the question?
Now, how you make sure that the selection of that particular gene is a stable thing? I mean, that it doesn't change.
How do we know that it's a stable? That's a good question. In fact, I don't think that this has been truly shown that it's stable. It's only been shown in the T-cell receptor, because in the T-cell receptor, the initial promoter choice is followed by a DNA recombination. So you have seen here in the protocaterins, you have these rings which link a certain promoter to the enhancer. So in T-cell receptor family, there is a similar looping, but afterward this loop is formed. The interceding DNA is simply cut out by a recombinase. So that makes it stable. So that's why the T cell, while in most other cases, it's, yeah, so I don't know whether, so how to say, at least in the odorant receptor, the separation between the expression and the non-expression state is so wide that it's difficult to imagine how a transition can occur there. But I think people simply did not show this, whether switching is fully excluded. But what is still a remarkable achievement, that how you can make sure that out of really, this is here, just we show here, I mean, not here, but from this review shows only 7 genes and one is chosen out of 7, but this is in fact from 1,500 genes. You have a single chosen and the others are suppressed. Mm-hmm. That is a, how to say, a really interesting stochastic phenomenon, and this is not solved. There is one model published some 15 years ago which has some tuning in it. I mean, so it's a classical stochastic model, and but they had to include a Hill number of 7, 8, which means that there had to be quite a lot of nonlinearity in order to, and there is some, in addition, some, because, so there are, so these are distributed, these 1,500 genes are distributed over multiple chromosomes, but along each chromosome, they are next to each other. So first, people suggest first a chromosome is chosen, and then within this one chromosome, one gene expressed, and then it suppresses all the others on the same chromosome. So it's a two-state process.
Mm-hmm. And of course, there is no guarantee that all the 1,500 are on in different sets.
But I think this is a system where stochastic gene expression makes a very, you know, strong impact on the biological behavior. That's why I like this system.
Any other question? I have only the last one. So instead, on the first part, I didn't understand this relaxation from the upper state when the gene is activated to.
Any other question?
Sorry, Attila, can you hear me? Hello?